Nice to see you.
Sarah, I
Uh,
Nancy accidentally admitted Nancy Kozak a guest while we're running tech. So Nancy, sorry, please bear with us. We normally wouldn't have let you in for the tech part, but we're waiting for Kara, who just came, and we'll get this going. And we should go live in 12 minutes. Hi.
uh, Hi,
Hey,
beautiful.
how's it
Hello.
going?
Good. How are you?
I'm good.
Good. Good to see ya.
Yeah, likewise. Can you guys hear me okay? I'll turn my volume up just a little bit. Can you hear me okay?
You sound good. Yeah. Uh-huh.
Great.
Yeah.
How
I was
are,
I was just wondering if you've done this talk for us. I'm sure it's not the same talk, but
no, so for this one, it's pretty much all focused on domoic acid and harmful algal blooms for the most part.
right,
The previous one, I touched on it a little bit. I kind of talked about like current straining events.
right.
I think because I went back and looked, I was like, I don't want to do the same talk. That wouldn't be. I
Yeah.
mean, I don't know, unless you wanted me to.
No.
But yeah, I talked about like the gray whale UME from last time, although here we are with a bunch of dead gray whales again.
Yeah.
And I think some neurologic disease. I did touch on DA. And there's something, maybe I talked about sea otters or something, something
Yeah.
else.
Something else. I wondered, I just was my only reason for saying that is just that Katarina brings Marine Mammal of the Month and it's sea lions, so that's really cool.
Oh,
And then,
perfect.
and then we have your minke whale that was in the bay. We processed the skull and it's just come out of the soak. So it was a
Oh, great.
past CMMC skull swap, and now we just brought it out. And it's really beautiful. And it was necropsy
Awesome.
down there, and I know the other one had DA. So anyway, if you want to touch on that at all, we're
Yeah, I did put a slide in there about the Minky since it was unusual. And I'm glad that's exciting that you have the skull already like cleaned up and
Yeah,
sort
we're
of
going to
prepped.
actually be
Yeah.
having a science event or a science social on July 9th that we should mention to the people who are joining us tonight as Mo Flannery will come up and talk about this seems to be an unusual mortality event and the minke whale and what we can learn from necropsies. So it'll be a
Good.
barbecue and a talk and a sneak peek at this beautiful skull that the Sarahs did
Wow.
an amazing job on. It looks I can't believe it. It's just made in April and looking at it now, I'm in awe. I
It
really,
was I wish I had like a video montage of all the efforts to get
uh,
out to that whale and then to like Porg was not gonna believe the tide was coming in. He's like, I'm getting this head, and we're just like, oh my God,
wow.
dude, we're all gonna drown out of here. But he was
Oh,
bound and determined to get the head, the brain. He obviously wanted the brain more than anything, as we do. But yeah, he was the fire department came back out and they were gonna help us, and they're like, do you need some help? Do you need like a big saw? We're like, yes. So I'm sure it was a little bit of a mess.
just a
Yeah.
little. She had some,
Yeah, it wasn't ideal. It was very deep mud, and it was a really difficult place to try to work in.
not to
Um,
mention rather difficult overall.
yeah, it's never euthanizing any animal always sucks, but a whale is it's definitely one of the hardest things I would say animals to ever have to euthanize. No
Yeah.
matter what, like
can I
it's
say,
just
Cara?
yeah,
Oh,
yeah,
well,
yeah. So yeah,
the
but
boric acid and sea lions is so in the forefront of everybody's minds these days. It's so many people are thinking about it. And I was just thinking about how lucky we are that we get to have you here talking to our
thanks.
people about it. It's just, what a valuable thing. And then it'll be up on our website and people can refer to it. And it's just it's a great resource for people. So thanks. It's
Oh, well, I'm glad I could oblige. Yeah, thank you so much for the invitation. It's always delightful to see you guys and talk and compare notes on everything that's going on in our areas because I mean, just all the overlap is crazy. And it's nice. It's just I'm always thrilled that we have this amazing network that talks to each other and works together. Yeah, we need it. We need all of it. We need everybody. So it's such a relief to know that we all have backup and that we have each other's backs. Like it means a lot.
yeah, well, and yeah, so and the big picture. I mean, we wouldn't
Yeah,
know that this is actually happening on the entire west coast of North America
yeah,
if we weren't all doing the work together. So what a
exactly. Doing the work, talking to each other, like doing our utmost to get like not just like a random sample here and there, but like good sample sets that you can really try to draw conclusive data from. Like that's critical. Like the bits and pieces, like they help and they add up, but to really get a big picture, a full picture, we need all of us and more.
yeah, and year after year. It's
Yeah,
yeah. Yeah. Well, do you want to run your stick your slides up to make sure that we are all
sure.
good? And so
Yeah,
we'll
absolutely.
introduce you and we'll ask our audience if they will help to support this with a donation and
Oh,
invite them to our talk or to our public events coming up. And
amazing.
yeah, is there anything that you'd like Sarah to highlight about you right now that in her introduction?
Not too much. I've been in the field for like 27 years or so. It says the participant screen is disabled at the
Oh,
moment,
okay.
but
So here we go. Yep, that's why they pay me the big money. Okay, where are you? There, okay. So I'm gonna make you a co-host.
okay.
Oh, yep. Okay, so now you're a co-host.
Okay, let's
And
see. Hopefully that will cooperate.
yeah,
All
there you
right. Can
are.
you see that?
Yep. Hmm.
And going into, is that presenter, is that the right mode? You can see everything?
Yep. Uh-huh. Yep. And I'm
Okay,
gonna spotlight you as
great.
well. No, I don't want to do that. I wanna spotlight the thing. So let's see. Um, okay.
All right. So yeah, so I think I'm just gonna go forward,
Yep. Okay.
try to go forward. There we
Okay.
go.
Great. Do you have any audio or video?
There's two videos that are pretty short, so I can go up to those. Oops.
Yeah, let's just try the sound on it just to make sure
Sure.
that the
I
sound will
may have turned the sound off,
work
at least for
better. Okay. Yeah, if you
one of
want to just
them.
talk over it, that's great. That's way better.
Are those videos? Oh, there's one. Yeah, I thought that might be easier. This one might, maybe I didn't turn this one down.
Where I record? Oh, there. Okay, we are recording already. Good.
Okay, I'm just gonna, this one doesn't have much sound, I think, in the background, but is that coming through okay? Like sometimes it's a little slow, videos are a little slow, but
Yeah, it
um,
looks like it's just kind of twitching and
yeah,
struggling a little bit. Oh,
yeah, yeah,
hard,
exactly.
hard.
Okay, great. Yeah, sad, of course.
Yeah,
But
but so
yeah,
important to
sad.
see.
The other one is somewhere. I thought I had one more. Oh, there's a sea otter. Let's see. There we go.
Wow. Oh my goodness.
Yeah, just a very sad looking otter. Okay, that's playing too for you.
Yes.
Okay, amazing. Okay,
Okay, great.
great.
All right.
So that should be good then.
Okay.
See. Cool. So do you want me to because you know me, like I can talk forever. Do you want me to stop like 10 minutes before the end
Yeah,
and
so our audience
and
usually kind of fades out at about 45 minutes or so.
yeah,
And we would love to have the opportunity to do some question and answer.
great.
Yeah,
Okay.
so is that okay for you?
Absolutely. Yeah, I tried. I have 47 slides and um some of the, like I should, yeah, so it should take about 45 minutes when I sort of went through it, but I tend to sort of elaborate as we go.
Mhm.
Do you want me to take questions as we go or at the
I
end?
usually actually kind of let you go through your flow without interruption unless, unless it's
Okay,
a small audience and then you can ping pong a little bit, but I like to let your focus and then at the end, I will
okay.
manage the chat or Kat will manage the chat or we'll do it together, however.
Sarah, would you like me to manage it or would you like to manage it? What works for you?
Why don't you go ahead? Why don't I just, yeah, I'll just take,
Okay, great. And
yeah,
you can
that
ask
was,
a couple of great questions at the end and I will as well
Okay.
if we have any. And yeah,
it's
Okay.
I
And
really,
if
know
anything
what you're
comes up,
most
like if
excited
somebody
and
like
interested in. So yeah.
if
Okay.
anybody needs like some clarification around anything, feel free to like just interrupt and be like, hey, just can you clarify or ask or whatever. Happy,
okay,
that's totally fine by me.
Yeah, and I'll
okay,
manage the chat along the way. So if somebody's asking a question or something, I'll let them know that, we
Great.
will be answering questions at the end and help to clarify as needed and possible. So.
All right. That sounds perfect.
It's interesting. We've only had a few DA animals up here, but at the end of March, we had Chai, which was a tagged female,
Mm.
come from PMMC, I believe,
Oh, interesting.
tagged, and she came up and she did a bunch of weird bobbing and collapsing on the beach, and then she took off. But she
Oh,
was beautiful body weight, and it was kind of a first for
good.
me to see an adult female. Um,
Yeah, that's she's kind of going the wrong direction.
Case in point,
Like,
what are you
are you
thinking?
trying to be a stellar? Are you trying to go back to Oregon? Where are you going, honey?
That was interesting,
Yeah.
but
Had she
no.
stranded at an we actually had we got
You guys, we gotta go,
oh
or
yeah,
it's
okay.
time to go,
Okay.
and I've got this roaring motorcycle now who loves to do that. So I'm gonna mute myself and let you guys take the lead on all of it and just admit
All
everybody
righty.
so we
Great.
don't. Okay, all
Good to
right.
go. All right.
Good evening, friends. And it won't overly surprise me if we have a few people still out in the sunshine because we've had June gloom for many days in a row.
Oh yeah, us too. It's actually I see blue sky and it's very exciting.
Exactly, exactly. So the beauty is that these are recorded and a lot of people take time to check in later. So and people will definitely come and watch that at a later time because they're all on our website. People can watch them, but this is a lovely audience so far.
Yeah.
Good.
Excellent.
And I don't know, well, we'll see. I think people will continue to funnel in. That's kind of the Mendo way. So we'll let people come on in and get settled. This is where I have fun and kind of put a shout out to the North Coast Brewery because they do support
Ooh, yes.
three organizations that are very near and dear to this work and to these folks. The
I have a magnet right next to my desk.
the Stellar IPA, put together by Mark Rudrick, our board member, as a way to support marine science, which, gosh, oh boy, howdy, what a great thing to support. And it needs it more than ever right now. So go buy lots of Stellar IPA for your friends and family. That's one way, drink for science. Um, It's a
I love
delicious
that. Great
beer.
for science.
Exactly. It's a really delicious IPA, in my opinion. The brewery dinner always highlights it. And anyway, it supports our speaker. It supports the Marine Mammal Center. It supports the Noyo Center, and it also supports Dr. Andrew Trite up in Vancouver with his work with stellar sea lions and beyond. So just another way to support these people that are doing incredible work. I'm going to go ahead and just introduce our guest tonight, and it is really an honor because I've kind of, when I was a little kid, I think I wanted to be Cara, but never really stuck with the long, incredible, intense journey that she has been on. Rather, I have been working in the field and sending patients to her, and just knowing that as a rescue volunteer for the Marine Mammal Center, that they're going to these incredible veterinarian science studied. We are learning about the health of the ocean through the work of Cara Field et al. So what a beautiful situation there. So actually, I'm going to hand it over to Kat real quick, and then I'll introduce Cara fully.
Hi, everybody. I'm Katerina. I'm the development director at Noyo Center. In a past life, I was the IT manager for the Society for Marine Mammalogy. So I met Cara staying at her house back in 2015 when we were planning a conference, and now we're here. And before we came on today, we were talking about how beautiful and important it is that we partner with Marine Mammal Center, and that we're up here monitoring and responding, and they're there, and we're all working together and doing this work. And in a time like this, talking about domoic acid and all of this important work that we're doing together, it really reinforces how important and vital our work is, and how meaningful it is that we've been able to do this for 10 years. So to do that, we depend on people like you. And so I'm going to post the link to ask if you might be able to donate a minimum of $10 tonight to help support our educational programs such as this one and keep the good work going of all of it. We would be so grateful to have your support and keep it all going. And I'm really looking forward to this talk. It will be posted on our website and available, and we will share it out. And so I think this is a hot topic, and it's good to see all of you here. So that's what I got. Thanks for your support, everybody. Links in the chat.
Thank you, Katerina. We're so lucky to have Katerina here on our team as well, growing us bigger, getting the word out. So as we were saying, Dr. Cara Field is an incredible, yet another incredible woman in science who I feel it's an honor to even brush shoulders with her. And she's shared some incredible moments with me and been there in difficult moments, some of the most difficult moments, and taking care of all these little marine mammals that we send down there, and then the learning is deep. So she is now the Director of Conservation Medicine for Marine Mammal Center, and going out and beyond in the bigger world of marine mammal conservation medicine and all of that. And speaking with a bigger voice internationally, nationally, and so forth, with her wisdom and all of that. So I feel lucky to call her a friend and a mentor, and just somebody who I really look up to. Yeah, I think that is enough from Noyo Center. We welcome Dr. Cara Fields. Please enjoy the talk, and thank you for being here.
Thank you so much, Sarah and Kat. You guys are making me blush. Thank you. And right back to you. It is an absolute honor to work with you and all of you, everybody within our Stranding Network and within our greater field of marine mammal medicine and science. It's truly a privilege to be with you today and to be a part of all of this. So thank you so much for the invitation and the introduction. So I will, hopefully that is that sharing okay? All right. So you might recognize some of these little friendly faces. And as you can see, we're going to talk about domoic acid today. Hot topic, obviously after the last couple months, and really honestly after the last 25 or so years especially. So you'll see next to my name a whole bunch of letters. So I'm a vet and a scientist and a board ed vet with a specialty in aquatic animal medicine. And really all of those letters means I went to school for a really long time, like graduated from 27th grade is how I like to call it. Because I love learning, always have. And there's always so much more to know. And it's truly been a journey of the more you learn, the more you realize you don't know. So it's really in this job that I get to work in. It's an amazing privilege to keep learning each and every day. So my title currently is the Director of Conservation Medicine, and you're like, what the heck is conservation medicine? So I'll try to explain that. It's basically this field that has emerged over the last few decades that where we really start to recognize not just individual animal or even population animal health, but we really start to think about how animals and people and the environment, how we're all linked so closely together. And all of us that work with marine wildlife and all wildlife really see that every day. We see the impacts of human activities, we see the impacts of natural activities and things. And we start to understand better and better that we're all linked together, we're all intertwined. Um, and the impacts are very broad, often from the things that we all do. So conservation medicine is an effort to keep kind of that big those big links in mind as we look at the small, medium, and large pictures. And I don't have to tell you all much about this because I think everybody is pretty aware of the Marine Mammal Protection Act, but I want to give it a plug anyways, because it's now under some threat. And I don't want to go political today, but I do want to just emphasize that this is a really important piece of legislature, along with the Endangered Species Act, that helps conserve and protect all of our marine mammals, and our endangered species, of course. And that this was a really unique and amazing bit of legislation that protects not just the individual animals, but the ecosystems and the habitats in which they live. And that's something that we're all rather concerned about right now, is a change, potential change in those protections, because we really have seen many species rebound after becoming nearly extinct or even thought to have been extinct, but others are still struggling. So just wanted to plug the MMPA there real quick. And I think I don't have to go into a deep explanation of this for all of you, because this is what you all are very familiar with. But just a quick reminder about what a stranded marine mammal is. And it can be a little bit harder to tell with our seals and sea lions. Obviously, any cetacean on a beach is not doing well. But seals and sea lions, sometimes they, of course, as you know, haul out and they rest and they have this terrestrial phase, so they can be a little bit harder to know exactly what their needs are. But these are the premises that we work under. So this is another slide that I don't probably have to tell you, but I just wanted to build on that emphasis, that we are a network. We all work together to answer questions about what is happening with our marine mammals and their sea turtles. We don't see too many of them up here in the central and northern California, but we see a few, and just highlight all the incredible facilities and individuals that contribute to our stranding network and response, all the way from the north coast of Oregon all the way down the state. And the Marine Mammal Center, as you all know, is kind of hanging out here in the red. And we have kind of three parts of our range: our northern range that goes through Mendocino, our sort of Monterey range, which is focused around Monterey Bay, where we see a lot of animals, lots of amazing wildlife in that region, and down to San Luis Obispo. And I did want to point out one geographic point, just to refamiliarize everybody with this, is Point Conception. Because this point in California, this little sort of inflection point in the Channel Islands, where we have our sea lion rookeries, our main sea lion rookeries, are really critical as we talk a little bit more about domoic acid. And as you all know, unless you haven't been turning on the news, and I can't blame you, this year has been a real struggle for Southern California with domoic acid toxicosis. So we'll get into a little bit more of that. And then again, Marine Mammal Center, we've been around for 50 years. I just have to give us a plug because it's our 50-year anniversary. So it's really exciting, and we're celebrating all of our parts of our range, of course, and all the amazing work that everybody has done. staff and volunteers for the last 50 years. It's been an unbelievable effort. We also have a small hospital in Hawaii for endangered Hawaiian monk seals. That's located on the Big Island. So again, just a huge thanks to many of you are also volunteers. And so we could not do it without you. That's how I got my start in this field as well as a volunteer for 7 years here. So it's an incredible group to work with. So I think again, like this is sort of just more background, response and rescue. We know that's our bread and butter. We see everything pretty much, have seen, do see, and respond to everything. A wayward pregnant elephant seal who's very, very angry and about to give birth, trying to cross the road. Not here, please, miss. Please turn back around and go back outside the bay. So everything from that kind of thing to entangled whales, to sea lions and seals and other critters in trouble. That's all part of what we all do. And no matter the cause, we are their voice and we are their helpers. And that's a pretty amazing position to be in. Since we have a nice large hospital here, we're able to take care of quite a lot of animals in a given year, anywhere between, we used to say 400 to 600, and that was kind of like 600 to 800, and now it feels like it's 800 to 1,000 animals a year. We do seem to see those numbers increasing. But we have a pretty full service hospital here. If you've never been here, we'd love to come and see you, have you visit, and show you how we take care of these animals. And how we diagnose them, like this little harbor seal with fishing hooks stuck in her mouth and in her stomach and in her flipper, pneumonia in sea lions. And we've got this incredible, talented staff in our diagnostic lab and our pathologists. It's just a really incredible operation. The other part of this that's so critical is teaching. So again, we have these amazing opportunities and we really value maximizing them. So taking what we've learned and the huge patient numbers that we end up with and making sure that we pass that knowledge on to up and comers, the next generation. So we take on a lot of folks and we're very happy to have them. Many of them are veterinarians or veterinary students or technicians, graduate students with different approaches to science and medicine, interns, professional developments, our colleagues from different areas, or maybe zoological institutions that don't have as many sea lions to learn from and about. And then, of course, our younger generations as well, our youth crew and our student and teacher programs and tours as well. So all of that outreach is so critical for all of us to maintain that level or try to increase the level of awareness about these animals and their health. And then, of course, the best, what we always hope to see is good outcomes from rehabilitation. So we want our patients to go back to the ocean if they possibly can. But our job isn't done there. That is an important milestone to reach, but it doesn't tell the whole story. It doesn't really give us any information about how they're doing out there. So in this bottom picture, you can see these two fur seals running back to the ocean. They both have satellite tags on, and they won't stay on too terribly long. But what we really want to know is, how are we doing? Are we doing the right thing? Are these animals going back to being normal animals, or is something else happening? So that post-release information is really critical as well to let us know really how we're doing and can we do things better or differently. Why do marine mammals strand? Again, you all know a lot about these. And this is a big question about how we're going to talk about domoic acid, because we've seen this presentation change so much over the years, and we've got a huge mixture of different issues out there, and a lot of animals strand with not just one thing wrong. So it's really incumbent on us to dig deep, to not just see an animal like this sea lion who's entangled in monofilament and just stop it. Okay, well, that's her only problem. Is there a reason she got entangled in that monofilament? Does she have brain damage from something else? Why was she so, was she just goofing off, going after fish in a net, or was she looking for an easy meal because something else is going on? So trying to understand those associations and relationships, investigating in relation to bigger events like oceanographic conditions and climate changes, and those kinds of questions always come up as well. So some folks are not really fans of rehabilitation, and that's understandable. You all probably know that Oregon pretty much does not do marine mammal rehabilitation, and other areas feel similar, sort of along the lines of we'll let nature take its course. Um, so there are risks to rehabilitating animals. Of course, we always want to release healthy animals back into the wild population. There are risks of altering pathogens like leptospirosis, for example, a bacterial infection that can cause kidney failure. If we treat these animals with antibiotics when they strand with that disease, are we going to release them with antibiotic resistance? Is that something that we need to consider what we're doing in the big picture as well as for those individuals? So these are things that we take very seriously, and we consider for every patient that comes in the door. And we also look at the big picture of what does this type of patient and their problem mean. But there are a lot of benefits as well. We learn a lot about these diseases, and all of you are very, very well aware of the highly pathogenic avian influenza threat that we have had over the last couple of years. And HPAI, or high path influenza, has taken out tens of thousands of marine mammals in South America, for example. It's killed off some harbor seals in Washington state, in the St. Lawrence in Quebec on the East Coast, and many marine mammals in other places are also being affected. So this is a nasty one. And yeah, we can find a bunch of dead animals on the beach, and we may be able to detect something or we may not. So taking in and caring for live animals as well as our dead animals is really important to be able to detect and understand what the clinical disease process looks like in these animals. And of course, this allows us to take additional samples that wouldn't be accessible to us to research and investigate further and even archive for future when we come up with these things that we don't know what they are, like domoic acid originally. It also allows us to hopefully correct some human-induced problems. Sea lions don't go around getting shot by other sea lions. Entanglements, those are strictly human-caused problems, no matter the species. So we do a lot, whether we realize it or not. So we have a huge impact on these animals, and this is an opportunity to maybe right some of those wrongs. We can develop novel treatments and responses, and we can apply those, even more importantly, to endangered species like Hawaiian monk seals, or Mediterranean monk seals, or vaquita, or other species. And there are a lot of other benefits as well. So I think just keeping in mind the balances of the benefits and the risks is really important as we go through this. And of course, everything that we learn just yields a vast, vast amount of knowledge. It builds on all this data that we all are collecting all the time. You can see from some of these publications, a lot of co-authors on there. That's because all of us work hard. And really, all of these deserve probably hundreds of co-authors, because it takes an entire community to see the animals, help them get samples, study them, understand what's wrong, and then report it out. So that's a massive process. And it's a really important one. Otherwise, we wouldn't know what we do at this point. All right, so that's a little bit of background, probably more than a little, more than you needed, but just wanted to give a little context there. So harmful algal blooms, or HABs, and I think everybody has heard of these. Everybody's heard of domoic acid and probably some other harmful algal bloom toxins, biotoxins. There are reports that these are increasing worldwide, and it's a little unclear, and we'll talk about it later, are these reports increasing because we're better at detecting them, or are these algal blooms actually increasing, or is it both? And they're getting to a point where we're starting to worry and see population level effects on some of our animals. And this year was no exception for that with domoic acid, with a lot of animals being affected, and realize the ones that we respond to, of course, are only the ones we know about. There are probably a lot more animals being affected and impacted that we never see. So just a reminder of what a harmful algal bloom is made of. We have phytoplankton, which are the most important link on Earth for providing oxygen and the base of our entire food chain. So we've got diatoms, which are silica-based phytoplankton. They grow really quickly. They have a really intensive nutrient uptake, and then they sink because they're a little bit heavier. And then we've got our dinoflagellates, which are a little bit slower growing. The flagellate is a little flagellum. They're deeper water nutrient feeders a little bit more, and they can migrate vertically in the water column, so up towards the light and back down a little bit. So we see both of these and some other organisms be part of harmful algal blooms. Over the last 30 years, we've definitely seen some long-term warming trends in our oceans. There are certainly some local and regional variations with that. And it's really important to keep in mind, of course, that these variations are natural. And this is, of course, part of the whole process of climate and weather. So just as a reminder, for example, so the El Nino Southern Oscillation, like right now, we're kind of in a La Nina year. You know, it's pretty cool. It's very productive waters. But we've had some really crazy years with some really warm waters here, as you can see during an El Nino year, and increased rainfall. So all of these climate and weather events have an impact on us, on the animals, and on the entire environment, and throughout these massive ocean systems. So some harmful algal bloom biotoxins that you may have heard of include some of these, and domoic acid, obviously. Saxitoxin is another one you may have heard of. Brevetoxin, and this is one, if you have visited Florida, especially in the summer, you may be very familiar with brevetoxin. This is sort of the organism that's associated with the true red tide. And those harmful algal blooms can be really, really bad. There's quite a few other types. I've only listed a couple here that are maybe a little bit less well known, but are out there and can cause maybe less severe signs in many cases, although anybody who's had really bad vomiting and nausea and diarrhea can attest to that not being fun at all. And then there are some freshwater biotoxins as well that we may see. So what exactly is domoic acid? So this is a question is like, well, that's a kind of a fundamental important thing to know. Like, what exactly is it? Why is all this hoopla? So it's a pseudonychia. It's a biotoxin that's produced by different pseudonychia species, and there are probably 5 or 6 or 7 different species, and probably even some that aren't pseudonychia that produce it. And their influence, so the production of these are influenced by biologic factors or biotic factors, so like nutrients and things like that, and then abiotic factors like weather and things. And so domoic acid is a neurotoxin, and it causes excitotoxicity. So here's a little depiction of what an end of a nerve cell looks like. So we have nerves, all our brains and our spinal cords and our hands and our everything. We have nerves everywhere. And it's amazing just how fast, like we think to move a limb or move a finger or move an eyeball or whatever, just how fast those nerves generate impulses to make something happen. So every time we move or think or do those things, we have these little receptors, and they have these different chemicals. And glutamate is this little guy demarcated here by these little black dots. They get released by one nerve, and they get bound up by receptors on the next little nerve, and that's how the signal gets back and forth and around. Well, unfortunately, DA or domoic acid is it's acting like glutamate. It's acting like this little neurotransmitter and its receptor, and it hangs out, it gets up into the body, taken up, and then it binds to the little binding site. And instead of just hopping on and off like the normal glutamate does, it hops on there and it stays on there. And then it keeps that cell being activated. So instead of those cells just like going, yep, 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 active, nope, yep, yep. It's just like active, 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 active. So the cells now they're constantly being active. So now they're running out of energy. So we have these little mitochondria, these little power centers in every cell that make energy. And when the cell runs out of energy, it starts to suffer damage. And that's usually called oxidative stress or oxidative damage. And with too much of that and no energy, that cell will die. And when one cell dies, a lot more may also die. So that's why domoic acid is so insidious, because it acts all innocent, like I just like kind of one of these guys, but it stays stuck on there bound and causes those cells to get overactive and basically subjected to cell death. So that's a real problem. We need our nerves. We need our nerves and our muscles and all of those things. So we have a lot of these kinds of receptors, especially in our brains and in our hearts. And obviously, both of these organs, our brains and our hearts, are pretty important for survival. I mean, we need them all. We need our kidneys, we need our liver, we need our spleen, and all those things. But those tissues might be able to suffer some damage and not be as bad. Our brain and heart, though, are pretty sensitive little structures, and they need this support, and they need their cells to be normal. So the end result is we can have ourselves some really severe neurologic heart and heart disease, and also gastrointestinal disease. But our intestines, our gastrointestinal system is pretty good at like pumping out new cells and fixing some of that damage, where our hearts and brains are often not nearly as adept. So that's why this is such a problem. So domoic acid, as you know, is produced by the pseudonychia, the algae. So here's a little silicon. So these aren't, I don't think these are pseudonychia, but they're silicates. So the little sardines and anchovies and all the other little fishes come along, they gobble these up. The sea lion comes along and gobbles up the fish or the human or whatever, and then everybody gets DA absorbed into their bodies. So that's how that works. How do we know that this is bad for people? Well, in 1987, there was this group of people that ate cultivated or cultured mussels from Prince Edward Island in Canada. And over 100 people got sick, and 4 people actually died. And these people experienced mildly, in the mild cases, they had vomiting, diarrhea, and some disorientation. And some had respiratory damage, some had became actually epileptic because of the permanent brain damage. And of course, fatalities are pretty bad. So as a result of this really it had been known that this compound existed, but it became a little bit more, people became a little bit more alert to the potential damages that an outbreak or that a large amount of consumption of domoic acid could do. So in California, we've had a pretty powerful monitoring system in place since 1991, including biomonitoring of all state fisheries. So all oysters and mussels, shellfish, and other critters as well that we eat. So that's all part of our, California's and other states, not just California, part of our seafood safety knowledge base. So what about sea lions? Well, the big sort of tipper was in 1998 when over 400 California sea lions stranded in May and June of that year along the central California coast. And these animals were showing signs that were very much like those people. They were suffering from seizures, ataxias, being off balance, just generally displaying abnormal behavior. And at the time, there were a lot of possibilities to think about that could be the cause of this. Was it a virus? Was it a bacterial infection? Was there some toxin out there, some biotoxin? And the monitoring shortly after this event started found that there was high concentrations of domoic acid in northern anchovies and low levels in blue mussels along piers. So the part here to remember is that anchovies are usually feeding a little bit further out in the water, and our monitoring systems are mostly sort of shore-based, right? So along piers and things like that. So these events may be happening out in the water, and we won't see DA on our shoreline for a little while. So after ruling out a bunch of other diseases and some testing, this became the first real report. And this sea lion, hopefully you can see the video a little bit, is definitely, you can see it's an animal in good body condition. She's in good shape. She is tremoring quite a lot. Her whole body is tremoring and twitching. She didn't have a grand mal seizure in this video, but that is a really common thing to see. And then actually there were some previous reports of marine mammals, including sea lions, having small clusters of animals having seizures back in 1978. And I think even going back further in 86 and 92. But this was the first time domoic acid was actually diagnosed as the cause. So since then, sea lions have been an incredible sentinel for us. And since that time, the Marine Mammal Center and many, many other collaborators have worked extensively to try to better characterize what the heck is going on with this disease. So in the couple years, about 5, 6, 7 years after domoic acid toxicosis was discovered in sea lions, a lot of time was spent trying to characterize what was happening clinically with these animals and what kind of damage it was doing. And so a big one, just like those people that had damage to the hippocampus, that was also noted and documented well in sea lions. So if you look down here, this picture here, kind of on the left, is a picture. It's a brain, and the brain has been cut open. And this structure right here that I'm kind of outlining is called the hippocampus. And this part of the brain is responsible for memory and, in some ways, emotion, and it has some other important roles. So for such a small part of the brain, it has a lot to do. So you can see this one, the little arrow is pointing there nicely for you. So this hippocampus is normal. It's nice and plump, and hippocampus basically means seahorse. So it's kind of shaped a little bit like a seahorsey, I guess. Whoever anatomically discovered that thought it looked like a seahorse, which I think that's fun. But you can see on this other side, on this other brain, this hippocampus is not happy. It is shriveled and shrunken, and there's a lot of empty space in here. So this is hippocampal atrophy, and atrophy just basically means scarred down or reduced in size. So this is a very unhappy hippocampus. And this is a common thing to see, is that sometimes we'll only see one side of the brain affected, and sometimes we'll see both sides. But you don't really know for sure. Obviously, we're not going to open the brain of a live animal, so this is a lot harder to diagnose in live animals because we can't get this close of a look. This is a heart from a sea lion that had domoic acid. And so up here are the large blood vessels. This is the right and the left side. And you can see this big area that looks really pale. And these other little areas here that are also really pale. So that's because those little cells that I told you about that bind to glutamate, the domoic acid bound to them, kept them activated, and all those cells were dying. So that's what's responsible for this tissue death and this atrophy. So as we saw more and more of these events happening, there was more and more need to understand what was going on, because humans, remember, we're still at risk as well. We like to eat our sardines and our anchovies and our other seafood and our shellfish. And we have sometimes to close those fisheries, as some of you probably like to eat Dungeness crab, for example. And you know very well that sometimes those fisheries are closed due to domoic acid levels being high. So by 2008, the epidemiology, like how these cases were presenting, it was changing. And now, instead of just these acute animals that could be treated for the seizures and released, we're now starting to see them not recover and have what's basically referred to as epilepsy. So they had permanent brain damage. And that was probably, like, we don't know exactly if that was due to the increased severity, increased number of blooms, and or other factors. And there was a big push at this point to also really work on correlating this brain damage, this chronic disease, and these acute events with environmental events as well. So since that time, we've continued to move forward and make a lot of progress in understanding more about how to deal with this, how to, on a big scale, as well as on the smaller scale, what are the outcomes? Like what's actually happening with these animals? If we return them to the wild before and they seemed okay, but now they're not, like we can't even return them to the wild because they're not recovering. They're staying in care, and 2 or 3 weeks later, they're still having seizures. Like, we can't release those animals. It would be wrong to release them to go out there and have a seizure and die at sea and drown horribly, or because they have sort of amnesia, for them to forget how to be a sea lion. So then there was a big push to put satellite tags on these animals and track them and find out where they go and what they do. But you need to know how badly that brain and hopefully heart is damaged to make that assessment. So now we turned to doing MRIs, or magnetic resonance imaging, in live sea lions, so that we could understand, like, okay, how bad does this brain look, or how not bad does it look? So this animal had, came in with seizures. Does the brain look okay? Okay, we'll put a satellite tag on it. Maybe the damage isn't notable yet, and we'll release it and see what happens. And then we started seeing additional other species also being affected. So northern fur seals, harbor seals. So this is an example of an MRI of a sea lion that had domoic acid exposure. And without going into great detail, this white part is increased, basically, flare or indicative of some kind of brain damage event. So the tissue itself is a gray part right here. Again, you can make out that little kind of hippocampus looking thing. And it looks much more normal on this side with just a small amount of this uptake of the contrast material around it. But this one has a big empty space. So that's one of those atrophied brain hippocampus. And it's not the one from this sea lion, but as you can see, this sea lion got a satellite tag and he was released, and he kept kind of coming back, and you can see him, he's kind of harassing the surfer a little bit, who, you know, it is the sea lion's ocean after all. But it's not normal, normally, for most sea lions to go out and harass surfers. Some surfers might say otherwise, but most of the time they don't. So we needed to understand what behavioral changes were happening with these animals so that we could better judge whether or not they're even releasable. And what kind of level of damage could they potentially withstand to still have a normal life or not. And furthermore, really importantly for us, they're a model of what's happening to the brains of these mammals that are feeding on this toxin. This is a unique, really naturally occurring model for what we could expect to see in humans, especially people that are consuming a lot of like that rely on seafood heavily. So consume a lot of razor clams, for example. A lot of people, that's their main source of food and protein. And if they are getting exposed to large amounts of domoic acid, we may see them coming up with the same exact kinds of issues, health issues, as sea lions. So they're a really important model for us to understand. So then some people also wanted to know, well, is this real? Like, do we now discover this and now we're just seeing it all the time because we know what it is? So interestingly, one of my brothers is a paleoceanographer, and he and his colleagues looked at basically 2,200 years worth of core sediment samples from the ocean floor from different parts. This one was in the Santa Barbara sediment sample, Santa Barbara Basin. And this is just showing the last, like, since 1975, some of these different Pseudonychia species. But if you look right here, you'll see in 1998, right before there is indeed a huge spike in the fossil record, basically, of Pseudonychia australis, which is one of those nasty domoic acid producing pseudonychias. And a real spike in these is found in those core sediment samples over the next kind of 10, 15 years. So this was published in 2008, but the samples went up to about 2006 or so. So this is real. The fossil record shows that this is not just, it's not just a matter of, oh, we got good at figuring out what it is. It's truly increasing. And so that's really important information to have. And then probably all of you remember the nasty, nasty blob or marine heat wave of 2014 and 16. And this was another kind of a bit of a game changer because we had unprecedented high levels of domoic acid and stranded animals. Here's the coast of North America here, and here we are going up and down the entire Eastern Pacific. And this year we really saw a lot of whales being impacted because of this, because the Dungeness crab season opened late. The whales were following prey and they were moving closer to the shore. So we saw all this increased number of entanglements. So that's sort of like a secondary problem. So economically, big problem for us because we don't have a crab season, and then a huge problem for the whales because they're getting entangled with their normal migration, the change in season. So this is just to kind of show you some of those shellfish harvests and fishery closures in 2015. You can see all throughout May, June, July, September, October, November, December, all year it just went on and on and on. And we had really high levels of domoic acid in places where we'd like barely seen it before. So at the Marine Mammal Center, we do our very best to diagnose this and document it. And you can see this is when we started documenting it in our patients in 1998. And then over the years, we've had some real spikes, some really nasty years. 2009 was pretty bad with about almost close to 300 animals coming in. There's 2014-15, also really bad. And then last year was really awful as well. So we got slammed with, again, about 300 or so sea lions that came in last year. And they're mostly coming in, if you look at this map, you can see this is patient numbers coming in here on the left and the month down here on the bottom. And you can see most of them don't really come in January. We do have some blooms that are a little bit earlier that kind of pick up in April. And this is actually where we are right now. This bright pink line, that's this year, 2025. But you can see like these blue lines, they spiked really early. So this blue line here represents 2015. But you can see like we got some pretty high numbers of D animals. And this is, again, this is just the Marine Mammal Center, not all of California. But it gives us an idea of kind of what kind of year we're dealing with on a bigger basis. So I mentioned earlier that Point Conception and the Channel Islands. So here's a little map of that. And this is really important as we go to this year. So as you all know, this was a really bad year for domoic acid in Southern California. And actually, so were the last couple of years. They've had a really rough time down there. So this is part of the oceanographic conditions. South of Point Conception, we see this is a Southern California bight, and it has a very particular oceanographic patterns that I am not qualified to talk about very well. But basically, these cold water upwellings bring a lot of nutrient-rich water up to the surface. And that nutrient-rich water with like nitrogen and phosphorus and all those elements feeds these algal blooms. And it feeds, of course, good algal blooms, but it also feeds harmful algal blooms. So that's kind of what happened earlier this year. So starting in February, these are the actual numbers. It's not a pretty graph, sorry, because they're still rough numbers. But here's all the way from San Diego up to Santa Barbara and Ventura counties, and then us here in our tip of our southern region in San Luis Obispo. This was really unusual to start getting, almost a couple hundred, about 150 animals in February. And then in March, over 800 animals, April, over 800 animals. And it didn't slow down until May. And this is really, really bad for a time of year. This includes both dolphins, by the way, and sea lions and birds. But this was a huge event with dolphins that happened this past year. So our little part here, so we're right on the tip. So again, here's Point Conception over here on the left. The San Luis Obispo County ends kind of like right up here. So we saw a bit of a tip of the number of animals that were coming in, but it was pretty rough out there. And so much so that there was actually, you don't usually get too many warnings about not harvesting Pacific sardines, but this one indeed was put into place, I think in March. No, in May, sorry. So why is it so bad? It was so bad that these flyers were issued, whereas February 21st with the first EA stranding this year, over 1,500 animals, 9 seafood advisories. So why is this event so severe and why is it different? So it talks a little bit about domoic acid, but it also talks about another one of our nasty biotoxins, if you remember, saxitoxin, which causes paralytic shellfish poisoning. So now you've got amnesic and paralytic, and they're both really, really horrible for these animals. So how do we know what animal's getting what? There's only one way to know, and you got to measure it. So that's what we try to do with both our live animals and dead animals, is get the best samples that we can to try to better understand what exactly is going on. Why was it so bad and so severe in February? We don't know. There is a theory out there that's floating around that the horrible fires in January in Los Angeles had a huge amount of flame retardant dumped on them. Flame retardant is composed mostly of water, but also is composed of fertilizer, which is basically ammonium phosphate. Both of those are food for algal blooms. So with the rains and wind and everything that drove more of this into the environment, there's a theory that these could have contributed and provided even more nutrients to help fuel these algal blooms this year. Right, there's no proof for that right now, and it is being looked at, but it is sort of a going theory on that. So darn you, saxitoxin. So it's out there and it's kind of always rumbling around. But remember, that's another dinoflagellate. It goes up and down the water column more. And we did also find this in California back in the 1920s with a lot of people getting really sick from it. And this one is really, really nasty because it targets these sodium ion channels and a few other channels, and it has a really high affinity binding like domoic acid, but to this different receptor. And it combines so tightly and so horribly that it causes respiratory paralysis and you die. So unlike domoic acid, this one is even more insidious with not a lot of leeway in the kind of ill category, but really a lot of mortality, a lot of death. And it has been associated with a number of cetacean mortality events over the years and in different species, in humpback whales, in North Atlantic right whales, short-beaked dolphins in Argentina. So it's definitely out there and it's been circulating for a long time. So how do we detect these things? How do we know what's going to happen? How do we predict it? We're very blessed that we have some pretty incredible technology to help us understand what is going on out there. We have these algal bloom monitoring systems using satellites, direct measurement, of course, with our little shellfish and our fish. There's modeling. All of these things help us understand what is happening. And so these are some of the amazing resources that we can use, the integrated ocean observing systems, to help us understand what's happening in basically in real time, and then correlate like that information. We can then correlate with the information that we get from our patients to help understand exactly what's happening. But we can only do that if we test for domoic acid, saxitoxin, and other stuff. And if we rule out other disease processes and pathogens like influenza, for example. So really important that we actually keep doing the work so that we can distinguish between what is a biotoxin, what is not. So when these animals started dying in February, all of us were like, oh no, like, yes, it could be domoic acid. It looks like it, but it could also be influenza because they look exactly alike, seizing animals washing up on beaches in large numbers. So there's a lot of different kinds of samples that we can get to look for domoic acid, urine, poop, stomach contents, a bunch of other things, and a lot of testing methods that we can use to look for it. Sorry, I'm going a little bit faster because I think I'm going too slow. And I warned poor Sarah and Cap that I would do this. Current treatments were really, really limited because there's no drugs that we can use to just bind up this toxin once it's in there. So we treat the patients with anti-seizure medication. So things like midazolam, basically like Valium, essentially injectable Valium, or phenobarbital, or a few other drugs. We give them supportive care. We give them fluids to hydrate them and flush out the biotoxins, anti-inflammatory drugs, because there's a lot of inflammation often associated with all that nerve activity, antioxidants to help protect those cells. And unfortunately, in pregnant females, that domoic acid goes down into the fetal fluids and then it recirculates throughout the body. So they often abort their fetuses when they're having these seizures. But if they don't, we actually induce abortion because we know that that pup is probably going to have brain damage when it's not even born yet, before it's even born. And that is not a fair life for that pup. And so we do what we can to at least try to help the mom. So it's a really, really horrible situation. And all of us get extremely frustrated when we have these cases. And that was one of the cases again earlier this year because a lot of the females that were stranding were pregnant. So it's a very, very challenging and difficult emotionally as well as physical work to respond to these events. So no treatment, unfortunately, to bind up this toxin. So the question, are harmful algal blooms associated with climate change? They're definitely associated with water temperature, nutrients, ocean acidification, because that's a fuel for different algae. Lots of things go into algal blooms, though. But overall, it does seem like the current conditions with ocean acidification and increased fertilizer and food for these blooms are producing more favorable conditions for these toxin-producing algal growth. So, and this seems now at this point to be pretty regional as far as whether or not they're increasing. They're increasing regionally. I think the data from just our region of California tells that story quite well. But they're also increasing. We're seeing more and more reports of them in other places like Alaska and beyond, of course. What about other critters? Absolutely. Unfortunately, our little sweet southern sea otters are also affected. I'm not going to run through their biology, but just mention that they're really critical species for coastal resiliency and kelp forest health because they eat back many of the critters, especially urchins, that destroy those kelp forests. So I'll go a little bit faster through this. This is a sea otter that is behaving very abnormally. I don't know if you can see this, but she's having a bit of a seizure here, paddling, twitching and tremoring in this little tide pool. And this is why it can be really hard to know how many animals are truly being affected by a bloom, because many of them will die out in the water and either sink and or get consumed by predators before we even know what's happening. So domoic acid is one of the many causes of strandings of southern sea otters. And there have been some great work establishing that heart disease or cardiomyopathy has been associated with a really high risk of death in southern sea otters. And this is really important to know. So it just highlights additionally some of those amazing partnerships. And this is potentially another population limiting problem for southern sea otters who have not moved beyond about 3,000 or so otters in over a decade now. And then, of course, whales. And so we come to the end. So I'm going to skip a little bit. You guys are all familiar with the recent events, but we've had a lot of gray whale mortalities kind of close on the heels of our previous gray whale unusual mortality event. And that's pretty alarming for all of us. In the previous one, there were some biotoxins found, but not really high levels. So that wasn't thought to be the cause of that mortality event. Instead, we see a lot of ship strikes. But what we don't know is, again, could this harmful algal bloom be affecting the whales and altering their behavior in a more subtle way that makes them more susceptible to something like a ship strike?
Cara, I just wanted to jump in here and tell you, take your time. This is
Okay.
incredibly rich talk. Do not feel rushed. If
Thank
people
you.
need to go, they can catch up on the recording later.
Okay.
But
I
feel
just
no
wanted to
rush.
make sure you guys had time for a
We're
question.
so we're so tuned in to what's going on with the whales and the work
Yeah.
TMMC and
Yeah.
Cal Academy are doing there in this overwhelming, and even the Southern California. So no hurry at all. Take your sweet time, girl.
Thank you. I'm actually almost done, so that's
Oh,
good. I just want to make sure I mentioned that we have, we're worried about domoic acid being part of the problem for our whales, and this is why necropsy is so important. Like we wouldn't know that these whales are getting struck by ships until we open them up and we see, a big area of hemorrhage, like you can see in this poor whale. This whale also had a fractured skull, broken ribs, and broken vertebrae, and there's nothing in the ocean that's going to do that other than a ship. There's no natural processes for that to happen. So unfortunately, this is what we're seeing again. Most recently, the last couple of months, we've had a lot of dead gray whales in San Francisco Bay and of course beyond. A lot of dead whales in Mexico again this year, at least 80, 85 or 90, I think were reported. And there's whales north of us also. So it's worrisome. And along with that, there have been a couple of other concerns because calf production is also pretty low for gray whales right now. So since 2016, the population has dropped quite a lot, back down to 16,000. I think it's even lower now. Sorry, I forgot to update that number. My apologies, but I believe it's currently estimated about 13,000 whales. But like this year, for example, in Mexico, I heard that calf production was very, very low. There are far fewer calves than there usually are. So this is very worrisome for the population. Like we don't know yet what this is going to mean for us, but it's something we're all keeping a very close eye on. And the role of domoic acid, saxitoxin, or other biotoxins and other factors is very much on all of our minds. So I just kind of wanted to close with this other bit of a sadder story. And this is about a minke whale that Sarah was telling me that you guys have recently cleaned the skull and prepped it, which is amazing. So I just want to tell her story a little bit. And we first heard about her right around the beginning of April. And Bill Keener, one of our awesome staff, was able to get these great photos. And this shows this whale. We're quite sure it's the same one. This is her blowhole, and you can see this really red, long, big discoloration on her back. And this was on April 2nd. And then she wasn't seen again, and then she was seen again on April 7th. So when we first saw these photos, my first thought was sunburn or a chemical burn of some sort. And we had no way to know or prove it because she hadn't really been sighted before. But then when we resighted her on the 7th, this is consistent with like all this, the dark skin has obviously sloughed off of her back, and now there's a couple little patches there. But it was really hard to tell. And she was still swimming, but she was seen getting a little bit stuck in shallow waters. And then unfortunately, on April 8th, she stranded in a mud flat very, very close to shore. And she swam all the way through. You can actually see the track that she swam kind of here in the mug. And she's on her side and in really bad shape. And once we got close enough to her, we could see that she was dying. And this was like she had all this incredible, this skin slough is covering a huge part of her body. And some of the other areas that you don't see here were infected and in really rough shape. So she was dying. She probably would have drowned as the tide came in, but we were able to get to her and euthanize her to save her any more suffering at the very least. And then at least we could get the samples to learn more about her and what was going on with her. So we did run Domoic acid levels on some samples that we got from her stomach contents and some feces and some urine. And we did find low levels, but we're not quite sure what that means because if she came into the bay and stranded, oh, sorry, I forgot to mention, there was also another minke whale in Southern California that was inside a little harbor that also died, and they did get high levels of domoic acid in samples from that animal. And then there were a couple other whales that also stranded that had high domoic acid levels. So we do think that this event has impacted these whales. It's possible this whale got exposed and just became maybe more disoriented and was affected by domoic acid. And by the time, a week, probably longer, she probably had not been eating. But maybe she had some residual because we just don't really understand what happens in whales. Presumably, it might be the same as sea lions and people and otters, but we don't know that for sure. And that's an area that we need to keep looking into more. So that leads us to our questions page. We have so many questions. We have a lot to learn about what's happening in developing animals, what's happening in cetaceans, what's happening in otters, how much DA is lethal, all these things. And so we all, we want to work on that together, and everybody helping answer these questions will help us get answers. And I know overall this is not a super positive way, it's kind of upbeat talk. So I do want to again reiterate just how important it is that we're all working together and collaborating to understand this health issue better and see if there's anything that we can do to try to mitigate some of this problem. Even if all we do is continue to monitor, study, and provide the supportive care, that's still doing something, and that still really matters. So I just want to reiterate the importance of doing that scientific research with our response and our rehabilitation to drive conservation medicine to help these animals and also help ourselves within that. Okay, so I'm so sorry, you guys. Thank you, Sarah, for that support. I definitely want to. I'm available if you have questions. I know that's felt like probably a speedy tour through domoic acid and hopefully not too confusing. But if anybody does have questions, I'm very happy to try to answer them. And I definitely do not know it all.
Cara, that was phenomenal. I mean, really beautiful description of Pseudonychia and the whole domoic acid. I learned something every time, and yeah, tremendously important. And it just hits home the importance of the work that all of these incredible volunteers, most of the people on the talk today are volunteers for NOYO Center and for the Marine Mammal Center, and doing that work where, just seeing how big it is when we actually do respond to deceased or ill or injured marine mammals. It's just, it's a great thing to be a part of and to learn so much from each individual animal. And I know our NOYO people are out every 2 weeks on the beaches surveying for dead and live and calling them in. And they're also part of the biotoxin sampling. So we do the mussels once a month, and we do from our pier and out offshore samples. And so do some people down in Point Arena as well. So great to be a part of this science, and more important than ever right now that we are collecting that science. So Sarah Quentin she's just saying awesome talk. Thank you. She's our local veterinarian, retired, but
All right.
Yeah.
Thank you, Sarah. Thank you both, Sarah.
Questions, please, if you want to put them in the chat, Katarina will read them. Questions, great time to ask questions. And if you want to be unmuted and just ask your question out loud, we have a lovely small audience here with lots of smart people.
While you guys are thinking up your questions, I want to invite you, if you're local, on July 9th, we're going to give you a sneak peek at the minke whale skull that Kara was talking about, and Sarah's have been processing it, and it's going to be ready to see. So come on over to the Noyo station, the field station, on July 9th. We're gonna have a barbecue on the deck, and Mo Flannery will be here from Cal Academy of Sciences talking about unusual mortality events and this Miki whale in particular. So it's going to be a really, really cool night. So do mark your calendars for July 9th. And yeah, and please do consider supporting our work at Noyo Center if you're able to with a $10 donation for this talk tonight. We'd be super grateful. And yeah, we'd love. Okay, we got a question.
All
Uh,
right.
Teresa Mercer asks, I'm wondering if as DA travels up the food chain, does it experience biomagnification?
Ooh, oh, that's a great question. Hmm. Um, I think at those lower trophic levels, that is what it seems like. So like we humans, like we might eat a whole bunch of mussels or something. And then it kind of that's it. Like we, if we get some of the biotoxin, it probably just circulates a little bit and we poop it out or pee it out or whatever. But biomagnification, so like kind of through the food web, I think a lot depends on the feeding habits of the individual animal, and that's probably how it biomagnifies or not. Um, so could we see, like, for example, so fish eating algae, sea lions and baleen whales eating, this bait balls of fish. And then what about an orca that ate a sea lion that just ate a whole bunch of sardines that were chock full of domoic acid? Now, is that orca gonna get domoic acid toxicosis? That's a great question. I don't think we have the answer to that question. Um, so I'm not sure if that really qualifies as biomagnification, but it seems like it's mostly related to the nutrients that are available. So I think it's, yeah, that's a, no, let me think. I guess how do you think about biomagnification in that way? I think it would be could be a short term kind of kind of event. Um, I don't think when the sea lions are eating the fish that are chock full of the algae that have domoic acid, yes, I do think that's like a more of a true biomagnification because the more they eat, the more they get. Um, but eventually it's limiting with how many fish have eaten how much algae with domoic acid. And remember, maybe I don't remember I said this or not, not every bloom is a biotoxin producing bloom. And that's actually something that we don't know what factors make this Pseudonychia make domoic acid at this time versus 2 weeks later, is that Pseudonychia still producing domoic acid? That's another really important question that we don't have the answer to. Um, so it is in one sense, but then I've never heard of like an orca eating so many sea lions that had so much DA that they got domoic acid toxicosis. But so from the mammal side, I would say probably not. But we also are lacking data, like good data in fish. So like if a shark ate a bunch of sea lions, they don't eat a bunch of sea lions though. So yeah, it's a really interesting question. Yeah, I think depends on how you think about that definition a little bit.
Okay, so Carl, I have a question. What are you feeling are the most fascinating and to you things going on in marine mammal science today? What's your what gets you up at 4 a.m. thinking about it?
Wow. Well, I have to admit,
You've got
my,
that eagle's eye view.
for a lot as a veterinarian, I have this weird balance. Like, as a veterinarian, I like to focus. I love focusing on the patient and what's in front of me and how can I help it? Like, how can I fix it? Because that makes you feel like you can do something. And that's a really good feeling to have. Like, am I doing something for this animal or in some way, shape, or form, even if I'm alleviating its suffering? Hopefully not every time, and it's not every time. but I tend to focus in those situations, tend to focus on the individual. But what I've really learned and come to embrace is the big picture effect, like population effects and species interactions and what's happening kind of on that big picture level. And that's something I've always like it's always kind of been there in the back of my mind, but more recently, those are the questions that keep me up at night. Is like, what is happening more broadly? And I am like, what is making it happen? Like, what are we doing? And what can I do to change that? And then it sort of fluctuates. If you guys have probably also experienced, like, how can I make a difference as a person, as an individual with what I do? And how do I convince people that this is real and it's a real problem and that we should all actually really care about it? Like I have relatives in Michigan, they're like, whatever, we don't live in the ocean. I'm like, that's not a good attitude. You should care about what's happening in the oceans. So like those kinds of things keep me up at night and kind of help drive that desire to keep learning and be engaged with the world, I guess. Like it's such an incredible place that these animals and the environment are so incredible. They're so precious. And um, I think I'm frequently just in awe of how cool and resilient so many of these animals are and how many secrets they still have. So those things kind of keep me up, keep me going.
Okay, so we got a question from our own awesome Sarah Bogard, who wants to know, or no, Sarah Bogard is from Manchester. Sarah Bogard came and gave us a talk about just recently about Harper seals.
Oh, cool.
Yeah, so Sarah says there was that article on coastal wolf in Alaska that brought up the risk of toxins from sea otter predation. Do you have any knowledge on that? Thank you.
Hmm. It's a great question. I think I remember hearing that the wolves were eating sea otters. Is that what you mean? They're consuming the otters and potentially getting some of their issues. Um, I know, I think we have not the same problem with wolves, but we do have coyotes that go down to beaches and they like to gobble up little harbor seal pups, especially, and chew on elephant seal flippers, it seems. Um, but yeah, so the Alaskan sea otters, their their disease sort of profile is quite different from southern sea otters. So our southern sea otters, we see a lot more like domoic acid, but we also see a lot more protozoal disease like toxoplasma and sarcocystis. But those can also impact northern otters. We did have like that distemper outbreak in northern sea otters at one point, but I think that's been a while ago now. I haven't heard that there's been another one. But that was a canine distemper. It wasn't seal distemper because we don't still, thank God, knock on wood. That's the next horror to come. We still don't have seal distemper on the Eastern Pacific. Obviously, it's on the Atlantic, and we see regular distemper outbreaks. But this was a canine distemper, so the otters probably got it from a canid because it was that type of morbillivirus, which is so distemper is basically, if you don't know, it's basically the dog equivalent or seal equivalent of measles, and everybody's aware of measles right now since we have that nasty outbreak. So yeah, so it can be even more lethal, canine distemper or seal distemper in dogs or dog-like species, canids, and in seals. So yeah, I other than that, I'm not sure which other diseases would be a big role or big concern for the wolves. I think there's trying to remember what else was in that sea otter, Alaskan sea otter mortality paper. And it's escaping me.
I have a question, and maybe we can, I'd love to further that conversation. I love that because we're all along this coast. But one thing that keeps me up at night is this interesting, what we see up here is like there'll be high levels of the algal blooms in further south. And we just for 22 years of rescue, how few domoic acid patients we've had up here. And these are free-ranging sea lions. So they're coming from down south and they're coming up here and they rarely present on our beaches. The females don't generally come up here, but the one that one of our volunteers reported on Noyo Beach was a female that was tagged. So her name was Chai. She'd been rescued and rehabilitated down south, treated for DA, and then was released and ended up here, which oh, was she not getting her directional signals right and then stranded? And we were going to do a rescue and she took off again. But that tagging tells us more, and then the sampling tells us more. And it's just so interesting that we don't really see a lot of DA up here. I was
Yeah.
expecting that we were going to get some higher numbers just because a lot of the sea lions were up here, but maybe they missed the bloom because they were already up here. But those are the kind of things that really fascinate me. But
Yeah, I totally agree. I think a lot of it is life history because the majority of the patients that we see with DA they tend to be adult females, and probably because they hang out around the Channel Islands taking care of their puppies for 9 months, 9, 10 months, and then they might branch out a little further and forage. But if that's where the bulk of the blooms are happening, then they're getting exposed potentially more and more regularly. Whereas the males are tending to forage a little further offshore. So when there are those big bait balls of sardines and anchovies, and a lot of the adult females are getting in on it, where the males might be more dispersed. And so I wonder if that life history part comes into it a little bit. And that's another thing that we unfortunately can't learn from these sea lions because we don't know, there's no way for us to know how much they've been exposed to and how frequently they've been exposed to it. So, like, do they have like a chronically low level of domoic acid and then just like, does it just like spike when they have a big bloom? Is it like just chronically built up till they hit a threshold where now they've got, permanent damage? Those questions are really hard to answer with wildlife when we don't know how frequently, how much, what are their habits like. Are the animals that are feeding on the schooling fish that are more likely to be eating the plankton, the ones that are more impacted? If you have loners or single animals, are they less likely to consume an entire bait ball? So maybe they're not getting as much of a toxic load. Yeah, wonderful questions that I wish I had the answer to.
So Karen Barreltimer has a question. What percent of the animals you see treat, in your opinion, are diseases caused by human, such as fishing lines, ship strikes, runoff, chemicals, etc.?
Oh boy, that's such a good question and such a hard one to answer because a lot of the time we do see those very direct effects like entanglement, gunshot, harassment. Varies a little bit with species and age. So we see a lot of harbor seal pups that come into rehab. It's because people interrupted that, mom pump on the bonding on the beach or scared mom off and, kidnapped the pup or what have you. So we do have like probably at one point we did a study and I think it said anywhere from 10 to 20% were like very directly associated with human. You could definitely put the blame on a human interaction of some sort. But there's probably a lot more than that because like with cancer in sea lions, for example, sea lion cancer is associated with a herpes virus, which is a naturally occurring herpes virus. But it's also increased risk of cancer because of pollutants. So how do you do the math on that? So when those pollutants are out there, these animals are acquiring it into their bodies, into their blubber, they're holding on to it for a long term. Those toxins and pollutants have effects on the body, on the immune system. So if it's reducing how good their immune systems are at fighting off diseases, like protozoal diseases or other diseases that may be more naturally occurring, is that a human impact as well? So where do we sort of like draw the line between what are all the human impacts that we can definitely say that animal's entangled in monofilament, that animal got shot in the head, those are human impact. But what about those animals that were made more susceptible due to much more difficult to diagnose underlying issues, like immunosuppression, for example? So that is a question, Sarah, to your earlier point, that question bothers me a lot. And it's much, much harder to prove cause and effect because we often don't see animals until they're stranded and near death. And at that point, it's pretty hard to prove like, oh, you got this or that disease because you're immunosuppressed. Um, so that's a really, really tough one to investigate.
I just love that we are getting those animals to the pathology and to you that we are learning. That to me is like the golden nugget of being a volunteer. It may have once been about rescuing a really cute little harbor seal pup, but it became quickly something that, we're not going to go out into those bait balls and watch them eat and count the fish that they eat, but we are going to look at their stomach contents and get them or rescue a big sea lion and get him down there and learn that he had cancer, urogenital cancer. Those
Yeah.
numbers are being studied, the domoic acid, the epilepsy, it's helping, it's human health, it's animal health, and that's all because of this important work that the largest marine mammal hospital in the world is just a mere 3 hours away from us. And I want to remind you, you volunteer for Noyo Center. Some of you also volunteer for the Marine Mammal Center, and it is an incredible team to be a part of, just doing that work and having this take home and massive amount of knowledge that we gain from these sentinels of the sea, as you said, and even just sea lions. Oh, there's so many of them, but we're learning so much because they are
Yeah.
such a
Yeah.
consistent sentinel.
Sometimes I'm surprised that that population hasn't totally tanked because
And it could,
I mean,
right?
between the Domoic acid, the cancer, the protozoal disease, the, all these other things, all these, food, lack of food resources with both normal but also probably human impacted in some way, shifts in food availability. Definitely some overfishing involved in that. So like again, that's that cause and effect. So how much that like I'm just so impressed at how resilient they are that their population hasn't completely tanked. Because around the time Marine Mammal Protection Act, they were being hunted, sea lions were being hunted for bounties made into dog food. So the population was somewhere around 50,000 or so then. Um, so I know some people still wish it was around 50,000. Others of us are excited that they've, bounced back, and they amaze me with how resilient they are when we see them come in droves like that. And you're just like, wow. Look at all these moms that are like in good body condition. They're nourishing these pups. Like it just impresses me so much.
Absolutely. Wow. Well, any other questions there in the chat? Anybody has anything else to add before we let Kara get out? Maybe catch a little bit of the sunset or catch up on charts or whatever.
Yeah, no. Fortunately, it was a quiet day on clinics today, relatively, so not too much there. Thank goodness.
We just responded to another dolphin. It was a new, I mean, she's stranded a month ago, but we were given access today, Rizzo, which was a
Oh,
first
wow.
for since I've been doing it.
Mhm.
Subadult female down on Manchester Beach. Sarah would have been here, my technician, but she was down there hauling the skull off, and it was code 5. It was very, very mummified and
Oh,
gone, but we had to wait till the plover nest nearby failed, and then we got to
yeah,
go monitor and get the skull and
gotcha.
pec
Wow.
fins. So
Yeah, no, deepest respect. I mean, every 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 single one of us who's out there like doing something to help improve our knowledge and what we gain, like every carcass, even if it's code 5, like there's still information we can get from that.
yeah.
It all really helps. It just shows how much we have yet to learn. So amazing. Like we keep at it. So huge thanks and kudos to everybody for participating, for being a part of this, for even just listening and sharing what you're learning, what you hear, trying to get your families to be involved. Some of our families are a little bit better than that than others, maybe, but yeah, any of those things, all of it all helps.
Well, we hope to have you up here before too long and
Yeah.
see the
Yeah,
fabulous
it's been far too long since my
we
last
can all
trek.
get our fan over with you and flies on the
Love
deck,
it.
nerd out on
Yeah.
animals in person. So please come visit. And
Yeah.
yeah.
It's been too long. I think it's been about a year and a half since my last trek up there. So yeah, I gotta swing by North Coast too. So yeah, all the things.
Absolutely. Yep.
Amazing. Well, thank
Thank
you again.
you, Cara. Thank you so
Absolutely.
much. Great.
Thanks.
Thank you to our
Have a good night,
volunteers
everybody.
and to Wendy and Peggy in particular for organizing these wonderful talks and to all of you for being here and for all that you do. I think, yeah, as Sarah has mentioned, that's pretty much everybody here has their hands quite involved in marine mammals, and what a thing what we're doing together. So high fives and thank yous and deep bows to all of you.
Go
Of course.
science.
Bye, guys. Have a great night.
You too. Thanks again, everybody.